All right, cool. All right, so let's dive in. Really quickly before we get started, if you guys have seen the lectures, then you're aware that this consent to audio or video recording release, we just have to go over really quickly. These lectures are recorded and they're going to be put on YouTube in a public forum. So if you're staying on here, by seeing this and by staying on the lecture, you're just agreeing that your information may be posted on YouTube. So if you have a profile picture or a name that's on your Zoom profile that you'd like to not have recorded, now would be the time to take that off, change your name or change your profile picture. But staying on just means that you acknowledge and consent to the recording. And right over here, this URL right over here, if you want to see the full release, you can always click that. It's on all of our lectures. There it is. Sorry about that. So this is the release. Just by staying on, you are consenting to it. Okay. So this is our 15-week MCAT course. I believe we're in week 12 right now. I put some links over here for you guys. There's the GroupMe link over there, our Instagram page. We have our YouTube page over there, which has all of our full lectures and sectional lectures, sectional segments from our lectures if you want to just look at specific topics. And we do 3 lectures a week for anybody that's new. We do them on Thursday, Friday, and Saturday at 8:30 p.m. And we have unstructured office hours every Sunday at 8:30 p.m. as well. So if you ever have any questions throughout the week about the material or just about MCAT prep in general, you can come. There's always going to be 2 tutors over there. And there will always be 2 tutors on each lecture. So my name is Chris, like I said before. I'll be presenting today. And then Janelle is also here. She's going to be the standby tutor. So if you have any questions, I would like you to keep your mics muted until the end of the section when I ask if you have any questions. In between, though, if you have questions, please feel free to use the chat box, and I or Janelle will answer them as soon as possible. Okay, so today's lecture is going to be mostly about metabolism, some of the pathways in metabolism. So we're going to start with glycolysis. And this is just a brief overview. I put this in here for you guys. We post all of the PowerPoints and all of the videos after the lecture, so you can always come back and look at anything that you missed. Okay, so quick introduction to cellular respiration. Humans oxidize glucose from start to finish through glycolysis, the Krebs cycle, and the electron transport chain. So the oxidation of glucose is the main form of energy that humans utilize. And oxidation is how we actually take energy out of the bonds from glucose. In the first 2 steps, not much ATP is produced directly. So in glycolysis and the Krebs cycle, we're not super concerned. They do produce ATP and GTP, but we're not super concerned with producing the ATP. We're actually concerned with creating the precursors and creating some of the molecules that will feed into the electron transport chain. So glycolysis produces 2 ATP molecules per glucose molecule, and the Krebs cycle also produces 2 ATP molecules per glucose molecule. So as glucose is oxidized throughout these processes, the electron carriers NAD+ and FAD are reduced to their high energy forms NADH and FADH2. So those are the 2 main molecules that I was just talking about that are super important for the electron transport chain. So really, we can look at these first 2 steps, glycolysis and the Krebs cycle. We can really look, their main function is to create these high energy electron carriers. So anytime anything is reduced, it becomes higher energy. So this NAD+, it gains an H with an electron over there, and it becomes NADH. So it's a higher energy molecule than this NAD+. And therefore, it's going to be able to donate those higher energy electrons in the electron transport chain. And it's going to power ATP synthase, but that is for another lecture. So glycolysis produces 2 NADH molecules per glucose. 2 spins of the Krebs cycle produces 6 NADH and 2 FADH2 molecules. So it's important to note that the Krebs cycle spins twice for every single glucose molecule. As we're going to talk about in a couple of minutes, by the time that singular glucose molecule makes it to the Krebs cycle, it is actually split up into two different molecules, 2 3-carbon chains. So these products, after glycolysis and the Krebs cycle, they feed into the electron transport chain. And in the ETC, 34 ATP are produced per original glucose molecule. So let's get into glycolysis. Glycolysis is highly conserved throughout all domains of life. And in fact, all living cells contain enzymes for the glycolytic pathway. So basically, all living cells are performing glycolysis as as the main way that they produce energy. So glycolysis has been around for billions of years. The enzymes are present in all different cells. It's a super simple way to do it. Obviously, animals and humans have evolved even more efficient ways to create energy, like oxidative phosphorylation with the electron transport chain. But pretty much all living cells throughout all domains of life contain the enzymes for this glycolytic pathway, so they do perform glycolysis. And glycolysis is split into 10 steps, and we can even divide those 10 steps into 2 phases. So the first 5 steps are known as the preparatory phase, and the last 5 steps are known as the payoff phase. Sometimes the preparatory phase is known as the investment phase as well, because we're actually going to be spending ATP in order to prepare these molecules for the payoff phase, where we'll actually make some ATP and we'll make some of those high energy electron carriers as well. And it's also important to note that glycolysis takes place in the cytoplasm. As we move over to the Krebs cycle and the electron transport chain, we're actually going to move to a different part of the cell. And the cell does this specifically. It sequesters these different actions in different parts of the cell just because there are different enzymes present in different parts of the cell. So glycolysis takes place in the cytoplasm, but it might not necessarily be possible in the mitochondrial matrix just because there are so many other things going on in the mitochondrial matrix. And vice versa. Maybe the electron transport chain or the Krebs cycle takes place by the mitochondrial matrix. It probably won't be able to happen in the cytoplasm because there are too many enzymes from other reactions that are, buzzing around the cytoplasm and interrupting. So everything has its place for a reason, and everything fits together kind of like a puzzle. So we're just going to go right into the steps of glycolysis. This is meant to be a brief review. I tried to put as much detail in it that I think is necessary for the MCAT. So glycolysis, there is a lot to memorize. I think it's a good idea probably to be very familiar with all the products and all of the enzymes. When we get to the Krebs cycle, I think the products are probably a little bit more important than the enzymes. But glycolysis, I think you should definitely be extremely familiar with and have a really good conceptual understanding of how it works. So we're going to dive right in. So the first step, a singular glucose molecule enters the glycolytic pathway when it is phosphorylated into G6P by hexokinase. So hexokinase is a type of enzyme. It's a kinase. And if you recall from other lectures, kinases, they phosphorylate is what they do. So they add phosphate groups to molecules. So hexokinase adds a phosphate molecule, to a glucose molecule. So G6P and hexokinase work on a negative feedback loop. An increase in G6P, which is the product of the reaction, will inhibit the activity of hexokinase, which is the active enzyme. So that's pretty simple to understand. We've been talking about negative feedback loops in a couple of our lectures. It's just one way that this specific part of the metabolic process is regulated. When G6P is upregulated, then hexokinase is inhibited. G6P is now unable to leave the cell, because of the negatively charged phosphate group. So obviously we have that hydrophobic phospholipid bilayer that's surrounding the cell. So now that there's that negatively charged phosphate group on there, G6P is no longer able to leave the cell, so it's sequestered. It has to stay in. G6P is also supplied to the glycolytic pathway by cleaving glycogen. So we have glycogen in our muscles as well. It's that sort of branched storage of glucose, and they're all sort of together. So when we cleave glucose molecules for glycogenolysis, they actually are released as G6P. So when you have G6P, it could be going to glycogen. It could be feeding into the glycolytic pathway. It could be a number of things. So if you look over to the left, I put this schematic. This is glucose. We see it requires an ATP over here, and hexokinase allows this glucose to turn into glucose 6-phosphate. So we look right over here on this 6 carbon, we have an additional phosphate group over here. So that's really the only difference between the glucose and G6P is this phosphate attached to this 6 carbon. And that's what hexokinase does. And again, it's important to note that this step is irreversible. So we have 3 irreversible steps in glycolysis. It's going to be step 1, step 3, and step 10. And we will go over that as we get to each individual step. But as of right now, I just want to make sure that you guys are following. We're just going to track this glucose molecule as it goes through glycolysis in the Krebs cycle. So we start with a regular glucose. We use up an ATP because remember, we're in the preparatory phase, the investment phase. So we're using that ATP and it makes a G6P molecule. And the only difference is that we attached right over here, we attach this phosphate group right up to the 6 carbon. Okay, so step 2, G6P is now converted into F6P. So we have glucose 6-phosphate. It is now converted into fructose 6-phosphate by phosphoglucose isomerase. This step converts the molecule into a usable form for the following steps. So step 2, we have G6P right over here. Phosphoglucose isomerase, it is an isomerase. It's the same exact molecule, just in a different conformation. You can look right over here. Fructose is a pentose, it turns it right into here, but the connectivity is basically the same. And that's all step 2 is doing. It is reversible. It's not like a committed step. It's not irreversible. It's just preparing the molecule for step 3. And step 3 is the really important one. So another phosphate group is added to the F6P molecule via the enzyme phosphofructokinase 1. It's often referred to as PFK1, and it produces a product known as fructose 1,6-bisphosphate. So this is an important step. You should definitely know phosphofructokinase, and you should definitely know fructose 1,6-bisphosphate. And like 1, it's irreversible. It uses up an ATP molecule because we're phosphorylating. And this is referred to as the committed step because this molecule can no longer be used in any other metabolic pathways like glycogenesis. So we saw step 1, hexokinase turned glucose into G6P. That's irreversible, but it's not considered a committed step because that G6P can still be used in other pathways, like we talked about, like glycogenesis. Once that molecule touches PFK1 and turns into fructose 1,6-bisphosphate, that is the committed step. This molecule is going to go down glycolysis, whether it likes it or not. And just a really quick note on nomenclature, this bisphosphate means that the 1 and the 6, bisphosphate means that the phosphates are attached to 2 separate molecules. If we had, say, a pyrophosphate, it would be 2 phosphates that are connected to each other, that are connected to one of the atoms. But the bisphosphate means that it's connected to 2 separate atoms. Yeah, so G6P turns into F6P down here, right at this diagram. And then phosphofructokinase, we're phosphorylating again. So we have right up here on this 6 carbon, we have that phosphate group, and then phosphofructokinase adds another phosphate group right on this 1 carbon over here. So it's important to just look at the slight differences because each of these steps really is just a slight difference between the others. So it's decently easy to follow. It's just all of the byproducts and the final product that are so different from glucose that we need to pay attention to. So we now have fructose 1,6-bisphosphate, and we can move on to step 4. So in this step, fructose 1,6-bisphosphate is cleaved by fructose 1,6-bisphosphate aldolase, which sometimes is just referred to as aldolase. And it's cleaved into 2 3-carbon sugars, and these are known as glyceraldehyde 3-phosphate. It's known as GAP, sometimes G3P, but it's actually cleaved unevenly. So the 2 molecules that are created are not, they're not identical. So one of them is GAP, this G3P we just talked about, and the other one is called dihydroxyacetone phosphate, or it's just DHAP. So those are the 2 3-carbon chains that are produced from fructose 1,6-bisphosphate, and this step is reversible. The remainder of the glycolytic pathway, I guess it's good to note, is just going to be oxidizing 3-carbon sugars. So from now on, when we're talking about 1 glucose molecule, it's going to be 3 carbons. We're going to have 2 molecules of 3 carbons. So that's why before, when I was talking about the Krebs cycle, how we need 2 spins for 1 glucose molecule, it's because we now have 2 molecules, and both of them are 3 carbons. Which obviously add up to the 6 carbons of the original glucose that we had. So like I said, it's a bit asymmetrical. So the gap that was produced can continue on to step 6. It actually skips step 5 because step 5 is just concerned with converting DHAP into gap. Because DHAP we can't really do anything with. We need that gap in order to move on to step 6. So step 5 is here. We have DHAP that is converted to GAP, and the enzyme that's responsible for that is called triose phosphate isomerase. Again, it's just another isomerase. We're just switching some atoms around, but it's basically the same empirical formula. It's the same atoms. So after steps 4 and 5, one glucose molecule has been broken down into 2 molecules of GAP. After this triose phosphate isomerase works its magic, we have 2 molecules of GAP. And I thought it was also important to mention that at equilibrium, after this step 4, when we have GAP and DHAP, and then we try to convert DHAP into GAP, the equilibrium of that is actually 96% DHAP. So it's super favorable in the reverse reaction. When we're trying to convert DHAP into GAP, 96% of that at equilibrium is going to stay as a product. So the reaction alone does not proceed forward very quickly. The reason why we're able to get so much GAP is because reaction 6 pulls it forward. So as soon as that GAP is created, it's going to be shuttled right onto reaction 6, and it's going to be pulled over to another product. So you have to sort of think about it like Le Chatelier's principle. If we have a bunch of stuff on the left of the equation, our products, and we have a very small amount on the right, when we take away from the reactants, when we take away from that small amount, we're going to start stressing the system, and it's going to shift over to the right. So it's going to start making more products. So we're actually pulling the products as they're being created because we're using them for other reactions. So that's pulling the reaction forward. And that's a common theme that you see throughout metabolism and throughout a lot of these sort of concerted systems, is that even if the specific reaction is not favorable, the reaction after it is probably very favorable. So it's going to be pulling it forward. All right, so we'll move on to step 6. Remember, our DHAP is now GAP, so we have 2 molecules of GAP right now, and this is now the beginning of the payoff phase. So we're finally going to start making some ATP. We're going to start making some of those high-energy electron carriers. And in this step, G3P hydrogenase, remember G3P is another name for GAP, catalyzes the oxidation of GAP to 1,3-bisphosphoglycerate. This is a reversible reaction, and this is the first step that actually produces the high-energy carrier NADH. So if we look up here, we're going step 6. We're converting, I think I actually cut off this product, but we're releasing an NADH, and the product is just we're adding this phosphate group right over here. So we have a bisphosphate again. Remember, it means that 2 phosphates are connected to 2 different carbons. So after step 6, we have right over here 1,3-bisphosphoglycerate. And we also just produced that NADH. So that NADH is going to be super helpful when we get to the electron transport chain. Step 7, we have this 1,3-bisphosphoglycerate, and the high energy phosphate on the 1 carbon, that one that we just put here, it's going to be transferred to ADP to create ATP via phosphoglycerate kinase. So again, remember, we're in the payoff phase now, so we're not using ATP to hydrolyze, we're actually creating ATP. So we take an ADP right over here, we take off one of these phosphates, boom, we get an ATP, and we get our product over here, which is 3-phosphoglycerate. So now we have this glycerate with only 1 phosphorus, with only 1 phosphate group. Yeah, and those are the products for step 7. Okay, we then have step 8. So right over here in this diagram, we're starting over here. This is our 3-phosphoglycerate that we just produced in step 7. And just keep in mind that we're looking at they're looking at a 3-chain carbon right now. So for every singular glucose, all of these steps after it's split, they're all happening twice. So we're only going over it once, but for each individual glucose molecule, it happens twice because we have 2 3-chain carbons, 2 3-carbon chain molecules. So step 8, 3-phosphoglycerate is converted into 2-phosphoglycerate by the enzyme phosphoglycerate mutase. So the mutase is just moving the group. The phosphoryl group on carbon 3 is removed, and a phosphoryl group is added to carbon 2, hence the name of the product molecule. This step is also reversible. So the only difference in step 8, we're just moving that phosphoryl group from 3 to 2. That's really it. And then we go to step 9. The 2-phosphoglycerate is dehydrated into phosphoenol pyruvate, which is usually just abbreviated as PEP, and it's catalyzed by the enzyme known as enolase. And this reaction is also reversible. And PEP is a super important molecule. We look at it in a bunch of different metabolic pathways, just going to and from, we look at it in gluconeogenesis as well, just the opposite way. But PEP can now enter the final step of glycolysis. So that brings us to step 10. Pyruvate kinase catalyzes the conversion of PEP into pyruvate by transferring its phosphoryl group to ADP to create an ATP. So the only difference between phosphoenol pyruvate and pyruvate is that we're removing that phosphoryl group. So we tack that phosphoryl group onto an ADP, and in step 10, we come out with an ATP and a pyruvate. So those are two products that we definitely want. Pyruvate is the final product of glycolysis, and it'll go through one more modification before it can hit the Krebs cycle. So the product pyruvate, immediately after step 10, is created in its enol form, but it quickly tautomerizes to the keto form because the enol form is super unstable. And the tautomerization pulls the whole reaction forward and renders step 10 irreversible once pyruvate is formed. So this is going back a little bit to what I was just talking about a couple of minutes ago. The original reaction from phosphoenol pyruvate to pyruvate is technically reversible, but because the enol form of pyruvate so quickly tautomerizes to the keto form, and the keto form is so much more stable than the enol form, it's not going to be able to flip back to the enol form, and it's not going to be able to reverse back into phosphoenol pyruvate. So that's why this step is considered irreversible. So I'm just going to repeat that one more time because it's kind of important. Phosphoenol pyruvate reacts with pyruvate kinase. It creates ATP, and it creates pyruvate, which is in its enol form originally, immediately. Once it is created, that enol form very quickly tautomerizes. That's the word that we use for transforming between the two forms. It immediately goes to the keto form. And it's very hard to switch back from the keto form to the enol form because the keto form is so much more stable. So because it's having so much problems getting back to the enol form, it's not going to be able to do the reverse reaction and turn back into phosphoenol pyruvate. So because of that, we consider the entire step 10 irreversible. So once we create pyruvate, it's not going back to phosphoenol pyruvate without the help of another enzyme that we're not going to get into in this specific lecture. Yes, and as Janelle said in the chat box, the keto form is almost always more stable than the enol form. Yep. So the two pyruvate molecules formed by glycolysis will head to the pyruvate dehydrogenase complex to be converted into Acyl Coa before the Krebs cycle. So Acyl Coa is actually what's spinning the Krebs cycle. So before we get to the Krebs cycle, we have to go through this one more step, basically, which is known as the pyruvate dehydrogenase complex. And we call it a complex because it's actually one big thing that's made up of 3 different enzymes. So E1, E2, E3. I didn't really get too in depth with it. You don't really have to know the specific names. E1, E2, and E3 are really sufficient. So after one glucose molecule makes it through the glycolytic pathway, 2 pyruvate molecules head to the mitochondrial matrix to the pyruvate dehydrogenase complex. So like we were talking about before, they translocate. They're at a different location than in the cytoplasm where we held glycolysis. So they go over to the mitochondrial matrix, and that's where this complex is. So in the PDC, pyruvate undergoes oxidative decarboxylation. So decarboxylation, if you don't know, it's just the release of a carbon dioxide from a carboxyl group. A carbon is released from the 3-carbon sugar as CO2, and the remaining 2-carbon entity is attached to a CoA group, coenzyme A, to form acetyl CoA. A molecule of NADH is also reduced to a molecule of NAD+ is also reduced to NADH. So PDC is crucially regulated by AMP levels or AMP levels. High AMP levels indicate low energy in the cell and therefore upregulate PDC activity to feed the Krebs cycle. So AMP is the result of when you take ATP and you remove a pyrophosphate. So pyrophosphate are 2 phosphate groups that are connected So we have adenosine triphosphate. When we remove 2 of those phosphates, we now have adenosine monophosphate. And it's a pretty good, when we have high levels of adenosine monophosphate, it's a pretty good indicator that our ATP stores are pretty depleted. So when PDC notices that these AMP levels are high, they're going to upregulate pyruvate dehydrogenase because we need more acetyl CoA to spin the Krebs cycle. So if we take a look at this diagram over here, pyruvate enters this first complex over here, and it releases CO2 because remember, we're decarboxylating it into a 2-carbon structure. TPP is the cofactor that this first pyruvate dehydrogenase uses, the first part of the complex. It then goes over to dihydrolipoyl transacetylase, and that's where the acetyl CoA is created. So it takes that 2-carbon chain, it takes the CoASH, which is just an abbreviation for coenzyme A. It's coenzyme A, and it actually has a thiol group, this sulfur right over here, and that's pretty important in terms, especially in terms of thialysis, which we'll talk about in a little bit. But the second E2, the second part of the complex, is where the acetyl CoA is released. And then the third part of the complex is where we take NAD+, we add an extra H and an electron, and we get NADH+ and a proton. Okay. All right, so before we do this question, does anybody have any questions? You can type in the chat box, you can unmute. I'll try to go over and answer them to the best of my ability. Okay, so then I will launch the polls. If you think of any questions, just put them in the chat box. Give me 1 second. I'll give you guys a minute or two, answer the questions. Remember, the polls are anonymous, so just take a guess. We're going to regroup in a little bit and we'll discuss. The polls should be live right now. All right, take another 10 or 15 seconds, get your answers in. Okay. All right, we're gonna end. Cool, so 20% said A, 60% said B, 20% said C, which is great because 60% of you got it right. So let's take a look at it. A molecule that started as glucose can no longer leave the glycolytic pathway after it has been acted on by which enzyme? So hexokinase, remember, acts on glucose in the first step. It is not the committed step. When it can no longer leave the glycolytic pathway, we're actually looking for the committed step. So G3P hydrogenase and phosphoglycerate kinase are not the committed step. G3P hydrogenase and phosphoglycerate kinase, at that time, the molecule is already committed. but we're looking at the specific enzyme that acts on the molecule that is the committed step, and that would be PFK1. So remember, it's that, that conversion into fructose 1,6-bisphosphate. And that's just basic like definition. Step 3, this is something you're just gonna have to memorize. Step 3 is the committed step, and it produces fructose 1,6-bisphosphate. Right there, yeah, so phosphofructokinase turns F6P into fructose 1,6-bisphosphate. So the step is irreversible, and it is the committed step. So once we have that 1,6-bisphosphate, we're not going anywhere. Okay, does everybody understand that? Okay.
Chris, I thought you said there were 3 committed steps. So why is the first one the most committed of the other 2? The other 3?
Yeah. So there aren't 3 committed steps. There are 3 irreversible steps. So step 1, step 3, and step 10 are all irreversible. They don't go in the reverse direction. Step 3 is the only one we consider the committed step, because that is the point where once the molecule goes through step 3, it's not going anywhere else. So every step after that, it's going to remain in the glycolytic pathway. But before that, the molecules in step 1 and step 2 are free to go in any other pathway that they want. So the question was just asking for which one is the committed step, which is the enzyme that is part of the committed step that will help us keep the molecule in the glycolytic pathway. So just because it's irreversible doesn't necessarily mean it's committed. Step 3 is the only committed step.
Thanks for the clarification.
Yeah, no problem. Okay. All right, so we're going to move on. We're going to talk a little bit about the Krebs cycle. I think for the Krebs cycle, the actual products are a little bit more important to know than the actual enzymes. And it's just important conceptually to know what the Krebs cycle is, what its products are, and basically what it's used for. So the Krebs cycle is also known as the citric acid cycle, the tricarboxylic acid cycle, TCA cycle for short. It takes place in the mitochondrial matrix. So remember, from glycolysis, we're coming over from the cytoplasm to the PDC complex is by the mitochondrial matrix, and so is the Krebs cycle. It's inside the mitochondrial matrix. So Acyl Coa is the product of the pyruvate dehydrogenase complex that we were just talking about. Pyruvate goes in, Acyl Coa comes out. This Acyl Coa can now spin the Krebs cycle. So it enters and it combines in the first step with oxaloacetate to form citrate. So I think I go in depth. Yeah. So we're going to talk about all of this. So if it seems a little bit overwhelming, don't worry. I'm just super quickly summarizing it. It combines with the oxaloacetate that's already in the cycle. This, if you look to the right, this Acyl Coa is coming in new, but this oxaloacetate is already in the cycle. So it combines, and the first step produces citrate. But basically, that's basically what the cycle is. They combine, they form citrate, then it goes through a series of steps where it's oxidized and it goes back to oxaloacetate, and then it combines again with the next Acyl Coa that comes in, and the cycle spins round and round. The Krebs cycle produces 6 NADH and 2 FADH2 and 2 GTP molecules per every original glucose. So remember, when I say original glucose, it's per 2 spins because there are 2 Acyl Coa molecules that came from that original glucose. So the glycolytic pathway has 10 steps. The Krebs cycle has 8 steps. So let's get into it. So the Krebs cycle, step 1, we have Acyl Coa coming from the pyruvate dehydrogenase complex. It enters the cycle. It combines with oxaloacetate and it forms citrate. Sometimes you'll see it as citric acid. I do believe at physiological pH, it's just citrate though. And the enzyme, I did put the enzymes on here just for your information. If you want to try to memorize them, you can. Enzyme for the first one is citrate synthase. And some of them, obviously the names are a little bit easier to, intuit where they fit into the cycle, and some of them aren't. So like the enzyme for step 1, citrate synthase, synthesizing citrate. That's pretty obvious. Step 2, aconitase, you might not know it unless you know the Krebs cycle. But like I said, we should focus more on the actual products than the enzymes for the Krebs cycle. So the citrate in step 2 is converted into its isomer, isocitrate. We're just moving it around. That's really it. Step 3, isocitrate is oxidized into alpha-ketoglutarate, which is a 5-carbon chain. And alpha-ketoglutarate is a pretty important molecule. It's an intermediate for the Krebs cycle. I think it's also used as a precursor for amino acids. So it's used in a couple of parts of your body. So it's an important thing to understand that all of these reactions that are happening, including glycolysis and the Krebs cycle and any sort of reaction that happens in your cell, they're happening in tandem with a bunch of other reactions that are occurring around them. So this is in vivo, which means it's inside of a living system. So there are going to be a bunch of other reactions that are happening all at the same time. So just because we're talking about something moving through the Krebs cycle, the next step, it turns into alpha-ketoglutarate, and then after that, it turns into Succinyl CoA. That doesn't mean that the alpha-ketoglutarate that was produced isn't going to go to another reaction. This is just, we're following it because we're following the Krebs cycle right now. But step 3, it's oxidized into the alpha-ketoglutarate, and it also produces a CO2 and an NADH. And the enzyme that is responsible for this reaction, or that catalyzes this reaction, is known as isocitrate dehydrogenase. Okay, so we now have our alpha-ketoglutarate. We let go of a carbon dioxide molecule, and we're going to move on to step 4. So it is now decarboxylated. Remember, that means we're taking a CO2, a carboxyl group, and we are releasing it again as a CO2 molecule. And it is a 4-carbon molecule now, and it's going to be combined with coenzyme A, and the product after step 4 is going to be Succinyl CoA. So with another CO2 and NADH form, just like step 3. And the enzyme that is used is alpha-ketoglutarate dehydrogenase, because we are dehydrating again, and it's a decarboxylation. So we release that carbon dioxide, and now we only have 4 carbons. So we're right over here. We now have Succinyl CoA over here, and then step 5, it is being converted into succinate. And here, the GTP and 1-succinate are produced. So it is a GTP that's produced. And the enzyme that facilitates that is Succinyl CoA synthetase. And it's important to know that the synthetase in this reaction is not actually synthesizing. It's doing the exact opposite. It's breaking down. So we now have succinate over here. So step 6, succinate is converted into fumarate as 2 hydrogens are transferred to an FAD. So we've been dealing pretty much with NAD for the Krebs cycle, NAD+. FAD is also that oxidized form of the high energy electron carrier. So after step 6, succinate is converted into 1 fumarate molecule, and we also now have 1 FADH2 molecule. So that is also going to head over to the electron transport chain, and it's going to facilitate with ATP synthase. And the enzyme here is succinate dehydrogenase, which also is a complex in the electron transport chain. But I'm sure we'll get more into that when we do that lecture. So we're right over here. We have fumarate. So now step 7, we're just adding a hydroxyl group to that fumarate, and it produces malate. And the enzyme that we use is malase. So you can see that all of these steps, we're basically just making little modifications to each of these molecules. Sometimes we'll cleave them, sometimes we'll add them, sometimes we'll just take off a functional group. And every time we do that, we're releasing a little bit energy, or we're transferring a little bit energy from, say, step 8. Say we have malate going to oxaloacetate. We're transferring a little bit of that energy to the NADH, to the NAD+ to create NADH. And then oxaloacetate is a little bit lower energy than malate. So that's where the energy is going. It's all about the energy transfer. And that step 8 is actually what regenerates the oxaloacetate, and that's what keeps the cycle going. So we started with acetyl CoA and a combination of oxaloacetate, and we're basically just oxidizing it all the way through the entire cycle until we finally get acetate again. And then once the oxaloacetate, excuse me. And once it is able to combine with another acetyl CoA molecule, we're just going to keep on doing it over and over again. And it's very beneficial. It makes a lot of those high energy electron carriers, and it makes some GTP also. Yeah. So the Krebs cycle creates FADH2 and NADH for the electron transport chain, but it's also responsible for supplying other biosynthetic reactions with reactants. So like we talked about before, alpha-ketoglutarate may leave the cycle to be converted into amino acids. And the cycle is regulated by many factors. So obviously, when you have this many moving parts, you're going to have a lot of different places where we can inhibit and where we can activate. So a couple over here, products of the cycle like NADH are going to directly inhibit enzymes in the cycle. So it's just that same exact thing that we always see where the products of a reaction are going to inhibit the enzymes of that reaction and slow it down. So when we have too much NADH, when we have a lot of these high energy electron carriers, the cell realizes that it doesn't really need to make them and it can conserve the energy and it can use it for other things. So it's going to inhibit the cycle. It's going to inhibit the enzymes directly. Same thing, citrate inhibits PFK1 activity in glycolysis because the citrate is going to be a product of the Acyl Coa, which is a product of glycolysis. So PFK is the committed step, so it's a pretty good place to regulate. It's a pretty good place to regulate the pathway. So if we have too much citrate, that means that we have enough. We don't really need any more materials to spin the Krebs cycle. So we're going to downregulate that PFK activity so that anything that's coming before, say we have that G6P, we can shuttle that off to glycogen and we can add it to our glycogen stores instead of making Acyl Coa and spinning the Krebs cycle for energy that we don't need. So your body is a very fine-tuned survival machine, and it's very sensitive, these sort of regulations. And calcium accelerates the link reaction. Link reaction is just another way to say the activity of the pyruvate dehydrogenase complex, and it upregulates the Krebs cycle. So if we have a high calcium concentration, we're actually going to upregulate the Krebs cycle because the pyruvate dehydrogenase complex is sensitive to calcium. All right, does anybody have any questions quickly before we go over the review? You can unmute or type them in the chat. So upregulating just means that we are increasing its activity. So if we're upregulating the Krebs cycle, we're going to be doing it even faster, even more. We have even more molecules. If we downregulate it, we're going to be inhibiting it. We're going to be stopping it. Okay, so I'm going to open the polls. Which statement is false regarding the Krebs cycle? If you think of any more questions, feel free to put them in the chat box. All right, polls are open, so take a minute and we'll regroup. All right, take a little bit longer. If the poll's not popping up and you want to access it, you can access it by hitting the poll button. It's on the bar. It's either at the top or bottom of your screen. It should pop up and you can do it. These polls are all anonymous, so take a stab if you don't know. All right, everybody get your answers in. We're going to close up in about 5 seconds. All right. Cool. So 100% of you said D, and that is absolutely false. True. So which statement is false? It's also known as the tricarboxylic acid cycle. That is true. It occurs in the mitochondrial matrix. That is true. It spins twice for every starting glucose molecule. That is true. and oxygen is required. That is not true. Oxygen is required in the ETC, it's the final electron acceptor, but the Krebs cycle does not require oxygen. We're just spinning acetyl-CoA and we're just modifying it, the citric acid. Okay, is that clear with anybody? Anybody have any questions? It seemed pretty obvious. Okay. All right, so we're going to talk a little bit now about lipid metabolism. This is the diagram for it. Again, I just put these here for you guys. I'll give you the slides after the lecture. And you can always come back and take a look, but I think I also put these on all of the slides after. Maybe not. Okay, beta oxidation is the first part of lipid metabolism we are going to talk about. So after being absorbed by the digestive tract, triacylglycerols, some of you might know them as triglycerides, are transported through the blood and lymphatic system to the organs in little lipoprotein vesicles known as chylomicrons. So those transport your fat throughout the body because obviously they are hydrophobic. Fatty acids, fat molecules are hydrophobic, so they can't travel through blood and lymph without some sort of vesicle or some sort of carrier protein. So that's what chylomicrons do. And once they get to the target tissue, the triacylglycerol is broken down into 3 fatty acids and then activated before it crosses the mitochondrial membrane. So when I say activated, I mean that acyl Coa synthetase converts a fatty acid into acyl Coa by hydrolyzing ATP to form AMP, as we were talking about before, and a pyrophosphate. So the pyrophosphate are those 2 joined phosphate groups. And acyl Coa, that ACYL, is sort of just a generic term that we're talking about because we're not really, that would change depending on how many carbons are in the fatty acid chain. So for example, if we had 16, it would be palmitoyl-CoA, acetyl-CoA is 2. So acyl is just the generic name for the fatty acid chain that's connected to the CoA. So that's what that means when we say acyl-CoA. So when an acyl CoA is in the mitochondrial matrix, a series of 4 reactions repeats on the chain molecule until the entire chain has been converted into acetyl-CoA molecules. So it's sort of like the Krebs cycle as well, where we're kind of just repeating. It's not a true cycle because different molecules come in, but we're just doing these 4 reactions repeated and repeated and repeated until we're only left with acetyl-CoA molecules. So the starting product of beta oxidation is going to be these triacylglycerols, and the end product is going to be a bunch of acetyl-CoA. So long-chain fatty acids must enter the mitochondrion using the Carnitine Shuttle. So short-chain fatty acids can just diffuse through the membrane. It's hydrophobic, but if they're small enough, they can just diffuse through. It's not going to be too much of a problem. But a lot of fatty acids are longer chains, and they do have to use this, this thing that we call the Carnitine Shuttle. So basically all it is we're just attaching it to a molecule, we're throwing it through the membrane, and then we're detaching the molecule and sending that molecule back outside. So the first step, carnitine palmitoyl transferase 1 transfers the acyl CoA to the hydroxyl group of the carnitine. So we have a carnitine molecule, we're taking the acyl CoA molecule that we really want, that we're interested in, we're just attaching it to carnitine and specifically on the hydroxyl group. So step 2, the new acyl carnitine is what we're referring to this molecule as, is transported into the mitochondrion via the transport protein carnitine acyl carnitine translocase. So this transport protein is an integral protein. It's in the membrane, and the acyl CoA cannot go through this translocase unless it's attached to a carnitine. So that's why we're attaching it. It's going to go through this translocase. Simultaneously, an unpaired carnitine molecule is transported out of the mitochondria. So it's what we call a symporter, or an antiporter, I'm sorry. One is going in and one is going out. So the carnitine, the acyl carnitine is going in because we want to get that acyl group in, and then just the unpaired carnitine molecule is going out because it's probably going to be used for another shuttle later. So that's sort of how we maintain that equilibrium of things going in and out. So once the acyl carnitine is inside the mitochondrion, carnitine palmitoyl transferase 2 converts the acyl carnitine back into acyl CoA and carnitine once it's inside the mitochondrion. So it's the palmitoyl transferase 2 does the exact opposite as 1. it just rips them apart. And then the carnitine is transported back out of the matrix, like what we just said in step 2, and the process just repeats, and that's how we get these fatty acids inside. So we're just attaching it to a carnitine, putting it inside for the translocase, ripping them apart, and sending the carnitine back outside. That's all it is. And like I said, the smaller chain fatty acids can diffuse through the membrane. Okay, so beta oxidation is actually a series of 4 reactions. As before, they act upon the fatty acid chain, cutting off 2 carbons from the end to form acetyl CoA for each cycle. So we have a bunch, the fatty acids, it's just carbons, it's just a carbon chain, that's all a fatty acid is. And we're cutting off from the end. That's what beta oxidation is. So acetyl CoA is just a 2-carbon molecule. So we're basically just modifying and cutting off these 2 carbons until we finally get down to only 2 carbons, and we just call it an acetyl CoA, and the entire molecule has been separated into 2-carbon acetyl CoAs. And it happens in 4 steps. So in the first step, Acyl Coa Dehydrogenase is the name of the enzyme, and it's used to remove a hydrogen and form a trans double bond between C2 and C3. So let me see, Acyl Coa Dehydrogenase. So we have this fatty acyl CoA right over here, right up at the top on this diagram, if you guys can see. It's coming in. So we have this Acyl Coa Dehydrogenase, and you can see the blue and red carbons. Those are the alpha and beta carbons that we call them. Once it's finished with step 1, we have this double bond right over here, and it's going to be trans because these 2 hydrogens are on the opposite sides. And we also create an FADH2 in the process. So that's good. So enoyl CoA hydratase then hydrates the double bond and adds a hydroxyl group to C3. So that's the second step. If we look right over here, we take this molecule that we just created, the enoyl CoA hydratase, adds H2O. So we look right over here at the beta carbon, this blue beta carbon. We now have a hydroxyl group and a hydrogen, as opposed to when we look back up before, we just had this double bond. So this was not saturated. We now have a completely saturated carbon over here. The hydrogen was also added back to the alpha carbon and added back to the other side of the beta. So we have a saturated molecule with a hydroxyl group now. And that's on C3, that's important to know. So beta oxidation, read that already. So the hydroxyl group in step 3 becomes a carbonyl in another dehydration reaction catalyzed by 3-hydroxyacyl CoA dehydrogenase. So we can look right over here. It's going to be another dehydration reaction. So we're going to be making, again, another carbonyl group. We're going to have a double bonded oxygen. So we go from this step over here. We're creating another NADH, one of those high energy electron carriers. And we now, on the beta carbon, have a double bonded oxygen. And like I said, NADH is produced. So this next step is thialysis, as I was talking about before. It's facilitated by thiolase. The bond between C2 and C3 is thiolized, and a CoA group attacks the C3. To reform acyl CoA, and that acyl CoA is now going to be 2 carbons shorter than the original because we just let go of an acetyl CoA. So we look over here, it's going to be broken right between this blue and this red, right between this alpha and this beta. It's going to split off. This R group just means it's the rest of the acid. So this left part is actually going to be the rest of the 16 carbon chain, say, if we had a 16 carbon molecule. So this all over here is going to remain. And these two, the alpha and everything to the right, is going to attach, it's going to separate. And this CoA molecule, it's going to become acetyl CoA over here. So we move up and we see that this carbon, this beta carbon that's blue, is now attached to a sulfur and another CoA. So we added another coenzyme A group on, but now since these two are gone, it's no longer a 16 carbon, it's now a 14 carbon. So that's basically what beta oxidation is. It's just those 4 steps being repeated. So it's going to repeat again as a 14 chain carbon. It's going to do the same thing. We're going to come right over here. We are going to dehydrate, excuse me. We're going to rehydrate with a hydroxyl group over here. We're going to dehydrate again, and then we're going to thialyze. We're now going to have a 12 carbon group. We're going to do the same exact thing. Another acetyl CoA is going to be released, and we're going to have a 10. And then it just keeps on going and going and going and going until we finally have 4 carbons. We split them in half, and both of them become acetyl CoA. And we just made a bunch of acetyl CoA from a fatty acid, and that is how beta oxidation works. And then the acetyl CoA can, move to other parts of the body, and it can be used to spin the Krebs cycle. Does anybody have any questions about that? I can definitely go back. Okay, and I am going to start the polls. Give me 1 second. Okay. So if we haven't explicitly gone over this, but you're definitely able to answer it if you think about it. So I'll give you guys a minute, maybe 2 minutes, just think about it. Again, the polls are anonymous, so I urge you to just, take a guess, click that poll button, it'll pop up. You can just put your answer in, and we're gonna go over it definitely. All right, get your final answers in. We're going to go over it. Nice. Okay, so I'm going to close the polls. So 20% of you said B, and 80% of you said C. So the correct answer actually is B. And I'm glad I finally stumped you guys because I felt like my questions were too easy. But we'll go over it. So how many rounds of beta oxidation would palmitic acid, which has 16 carbons, undergo before it's completely degraded? So if we think about it, we have 16 carbons. We cut 2 off. We now have 14. That's 1 round. 14, we cut another one off. We now have 12 carbons, 2 round. We cut another one off, 10 carbons, 3. 8 carbons, 4. 6 carbons, 5. 4 carbons, 6. And then we're going to do 1 more cut between those 4 carbons for 2 acetyl groups, which makes 7. So that's a pretty good thing to know, is that anytime you're doing beta oxidation, it's going to be 1 less than half of the amount of carbons. Just because that last one, when we cut, we don't need to cut anymore because that acetyl CoA is just going to be there. So the formula, like the formal formula, would be N divided by 2 minus 1. So we have 16, where N is the number of carbons on the fatty acid. So we have 16 carbons. 16 divided by 2 is 8. you might normally think that it's going to be 8 rounds of beta oxidation, but you have to -1 because that 7th oxidation, what's remaining is just going to be 2 carbons. So we don't need to do another round of beta oxidation. We just have to tack a coenzyme A on there, and we have our Acyl Coa. Does that make sense to everybody? Because I can definitely explain that again. The formula is N/2 - 1. I'll type it for you. Where N is the amount of carbons. So it's just half of the amount of carbons minus 1. So we have 16 divided by 2 is 8, minus 1 would be 7. If we had a 12 carbon fatty acid, it would be 12 divided by 2 is 6 minus 1. So it would undergo 5 rounds of beta oxidation before we could get those acetyl CoAs. All right. Great. Okay, so we talked about oxidation, which was the catabolic process. So now we're going to talk about fatty acid synthesis really quickly. So catabolism takes place in the cytoplasm. The oxidation is a form of catabolism. It's fatty acid catabolism. But oxidation, the anabolism synthesis, takes place in the mitochondrial matrix. I think that might be. Anabolism is not oxidation. Catabolism is oxidation. So I'm going to fix that. Yeah, but we're talking about anabolism now. So anabolism is fatty acid synthesis, and it's going to take place in the mitochondrial matrix. So the first and committed step of fatty acid synthesis is the conversion of acetyl CoA to malonyl CoA, and it's catalyzed by acetyl CoA carboxylase. So the first step of fatty acid synthesis is its committed step. So once that happens, it's turned into a fatty acid, whether it likes it or not. And that's right over here. So we have the acetyl CoA. CO2 is actually taken in. ATP is hydrolyzed into ADP, and it also uses biotin. So acetyl CoA carboxylase. I believe biotin is the cofactor for acetyl CoA carboxylase. And all we're really doing, all we're really doing is adding this right over here, just this phosphoryl group right over here. We're just adding it to acetyl CoA, and that becomes malonyl CoA. So that's the first step of fatty acid synthesis. So does everyone see that it's sort of like the opposite of decarboxylation? We've sort of been going the other way. We've been taking these carboxyl groups and taking them off as a CO2. Now we're taking in the CO2 and we're adding a carboxyl group and creating malonyl CoA. Okay, so fatty acid synthase is a very large enzyme that has multiple catalytic domains. The binding domain is known as the acyl carrier protein. Acetyl CoA binds to the ACP first. So that's sort of like the first domain. It's the binding domain. So we have a molecule. Remember, we made the malonyl CoA, but we also still have acetyl CoAs that are just floating around. So acetyl CoA is going to bind to the ACP. And then it's going to be shifted over to another domain and held in place by a cysteine residue. So remember, the acetyl CoA, coenzyme A, has that thiol group, it has that sulfur group. So it can react with cysteine residues. It's going to make a disulfide bridge, and it's going to hold it in place pretty well. So it binds to the ACP, and then they shift it over to another domain that has a cysteine. So it's sort of like shifting it over into a little, like a holding place. So it's going to hold that acetyl CoA. Then we have malonyl CoA that's going to bind to the ACP, that binding domain. So we now have malonyl CoA and acetyl CoA right next to each other. So we're going to condense them together. And in that condensation reaction, the carboxyl group from malonyl CoA is going to decarboxylate and be released as a carbon dioxide. That's where the energy of the reaction comes from. And the 4-carbon chain is then reduced twice by NADPH. We've been talking about NADH, but NADPH is also a high-energy electron carrier. It's created in the phosphate pentose pathway, pentose phosphate. And it shifts over to the cysteine domain. The 4-carbon chain is reduced, shifts over to the cysteine domain, and then it just repeats the process again. So another malonyl-CoA enters the ACP, and the process restarts. We're just condensing, decarboxylating, and then shifting over to the cysteine domain to hold it. So it basically is just beta oxidation in reverse. And this is the schematic that I put to the left. ACPCO2, we're removing carbon dioxide. We're going to condense the acetyl-CoA and the malonyl-CoA. And then we have these two reductions with the NADPH. So it's going to reduce. There's another dehydration reaction in between. It's going to reduce. And then this is our product. We are now 2 carbons longer for our fatty acid. And the same thing, it feeds right back in. So it's just a cycle. As you can see, a lot of metabolism is just cycle, just because it's super efficient. And the process repeats until a 16-carbon fatty acid is produced. So it always goes up to 16 carbons. It produces palmitic acid. That's your finishing fatty acid. We're done. Okay, we're going to talk really quickly about ketogenesis. So ketogenesis occurs during periods of starvation when blood glucose and glycogen stores have been depleted. So obviously, carbohydrates are the body's preferred source. Glucose is the body's preferred source of energy. So when we can't get it from the blood, we look to glycogen. When we can't get it to glycogen, we got to look somewhere else. So there are a couple of other things that'll happen. Our body will start gluconeogenesis, but ketogenesis also increases because we don't have any fuel, basically. So when this happens, the mitochondrial matrix produces what we call ketone bodies, and the process in which they are created is known as ketogenesis. So ketone bodies, the really important thing about them is that they are able to cross the blood-brain barrier, and they can provide the brain with energy when glucose is absent. So that's really important. The brain loves glucose. It is definitely its preferred source of energy, but when there's no glucose present, there's really nothing you can do. So the body switches over to make ketone bodies, and it tries to get them right up to the brain. So they are transported into the brain, and it's converted back into acetyl-CoA before entering the Krebs cycle. So we don't even need to do glycolysis because obviously there's no glucose. We make the ketone bodies, it gets to where it needs to be. It's broken down into acetyl-CoA, and we can start spinning the Krebs cycle again. We can start making NADH, FADH2, and we can move those over to the electron transport chain to create some ATP. The final products are 3 ketone bodies. It creates 3. Beta hydroxybutyrate, acetoacetate, and acetone. It actually first creates acetoacetate, and then the acetoacetate can be transformed into either beta hydroxybutyrate or acetone. Beta hydroxybutyrate is actually the most common ketone body that you'll see throughout, and acetone is the least common. But acetone is actually responsible for when diabetics go into ketoacidosis and you actually can smell like acetone on their breath. It's just because they're in such a state of ketoacidosis that even though acetone is the least common ketone body, its presence in the blood is so high that we're actually breathing it out because it's very volatile, so it's going to come out in your breath. Ketone bodies are negative, so that's why it contributes to ketoacidosis. It lowers the pH in the body, and then all of our compensatory mechanisms are going to start. We'll hyperventilate. We're going to breathe really fast because we're trying to get the carbon dioxide out of our body to increase our pH. Our heart rate is going to increase, all of that stuff. So ketogenesis is regulated by the availability of carbohydrate energy sources in the body and how it affects the acetyl-CoA levels. So the carbohydrate availability will affect acetyl-CoA levels, and the acetyl-CoA levels are really what's going to affect the ketogenesis. So when the body does not have free carbohydrate fuel, fatty acid oxidation creates an abundance of acetyl-CoA to compensate. So that beta oxidation is going to be creating a bunch of acetyl-CoA because there's no free carbohydrates. So beta oxidation is just going to be going because it's trying to make that energy. Under normal conditions, the acetyl-CoA would feed into the Krebs cycle to produce higher energy precursors for the ETC. However, when ketogenesis is occurring, it's going to be because of starvation conditions. So under these starvation conditions, the intermediates from the Krebs cycle, mainly oxaloacetate, they're going to be depleted because of the activation of gluconeogenesis. So a lot of the a lot of the intermediates in the Krebs cycle are also precursors and intermediates in gluconeogenesis. So when we're under starvation conditions, we're going to shift our resources from the Krebs cycle to gluconeogenesis because we really need to make glucose. We have to get our blood glucose up. So that's going to take priority over the Krebs cycle. And because that's happening, even though we have this abundance of Acyl Coa now from this oxidation, we're not going to be able to spin the Krebs cycle as efficiently because we don't have everything we need. It's all going to gluconeogenesis. So this is actually driving the activity of the Krebs cycle down. It's down regulating the Krebs cycle. But this high level Acyl Coa is actually what activates ketogenesis. So those Acyl Coas are going to be turned into ketone bodies. Does anybody have any questions about that, about oxidation, ketone bodies, ketogenesis? I didn't go too in depth about ketogenesis. I don't think it's like super high yield. Again, like a lot of these are super in depth. There's a lot of detail that you can know. I think the most detailed that you should study is definitely glycolysis, and be very familiar with the Krebs cycle. But if you do have any questions, you can unmute, you can put them in the chat, because we do have a few more questions, a couple of more. Okay, so I'm going to start the polls. Give me 1 second. All right, polls are open. Take a minute. All right, take a few more seconds, get your final answers in. Okay, I'm going to stop the polls. So we had 20% of you say A, nobody said B, 80% of you said C, and nobody said D, and nobody said I don't know, which is cool because the correct answer is C, in fact. So let's look at the question a little more closely. A type 1 diabetic experiencing an episode of ketoacidosis would most likely exhibit all of the following symptoms except, and it's not really super important that you even know the difference between the two types of diabetes, just the fact that diabetes leads to an increase in your blood glucose levels. So they would exhibit all the following symptoms except low intracellular levels of glucose. That would be true because all of their blood is basically sequestered. I mean, all of their glucose is basically sequestered in the blood. So whether it's because there's no insulin present or because there's an insulin resistance for the cells, regardless of either type of diabetes, that's not going to be true. Excuse me, that is going to be true. There's going to be low intracellular levels. So B, an increase in acetone and other ketones found in her breath. That's also going to be true. Like we just spoke about that a few minutes ago. Ketoacidosis, when we can't get fuel into our cells, we upregulate ketosis. We go into a state of ketosis, excuse me. So acetone and other ketones are going to be very highly concentrated throughout our body, and they're going to be very highly concentrated in our breath. D, an increase in ketogenesis would also be true. If it weren't true, then B wouldn't be true because we don't have that fuel. C is incorrect because high levels of glucagon secretion from the pancreas would not occur. Glucagon is secreted when there are low blood glucose levels, and we would not expect a person experiencing ketoacidosis to have low blood glucose levels. They will probably have very high blood glucose levels, and they just can't get the glucose into their cells. Does that make sense for everybody? Okay. All right, moving on. We are almost finished. We just have a few more topics to get through. So we're going to talk very quickly about insulin, glucagon, and metabolic regulation. Okay, so insulin is a peptide. I'm sure we're all familiar with it at this point. It's a peptide hormone created by the beta cells of the pancreas. So the beta cells are found in clusters of specialized cells known as the islets of Langerhans. There are actually 3 types of specialized cells. We're going to talk about 2 of them. And I would argue that they are the 2 more important ones to know for the MCAT. So insulin mainly serves to lower blood glucose levels by allowing its uptake into surrounding tissues. So the way that works mainly is that your adipose tissues and striated muscle tissues, basically where we need glucose to go, they contain transporters that are known as GLUT4 transporters on the membrane. So these are insulin dependent, and they will not take up glucose in its absence. So these are, like I said, they're on all of your striated muscle tissues, they're on your adipose tissues. Glucose cannot pass through unless insulin is present. So that's why there's a problem with type 1 diabetics when their beta cells aren't producing insulin. There's no insulin to allow the glucose to pass through the GLUT4 transporter. So it's not going to do anything. And with type 2 diabetes, there's actually a deficiency with the receptor on the GLUT4 transporter. So we have insulin, but it's just the GLUT4 transporters are not reacting to the insulin. So slightly different mechanisms for both of the diabetes, but it's basically the same thing. The GLUT4 transporter is not getting the insulin in the way it's supposed to, and it's not allowing glucose to transport up. And there are different types of GLUT transporters. So like, for example, the liver is lined with GLUT2 transporters, and those are insulin independent. So those don't need insulin. The liver can uptake glucose without insulin. It doesn't have a very high affinity for glucose, but whenever glucose does touch the GLUT2 transporter, it can be transported in without insulin. So the release of insulin also has metabolic regulatory effects. It doesn't just allow glucose to uptake into cells, it also affects our metabolic processes. So it increases fat synthesis, it decreases lipolysis, it's going to increase glycogenesis, it's going to decrease glycogenolysis, and it's going to decrease gluconeogenesis. So to summarize, insulin basically increases anabolic activity in the body and decreases catabolic activity, which makes sense. So you know that after you eat a big meal, your insulin levels are going to spike. So insulin levels spike when we are well fed, when we have access to adequate energy. So when we have a bunch of energy coming into our bodies, your body's going to release that insulin, and it's going to start these anabolic processes because it wants to conserve energy. We have all this energy over here, so we don't really need to be we don't really need any catabolic activity. We don't need to be breaking down glycogen. We don't need to be producing glucose because we have all of it. So what do? We want to store it. So when insulin is released, it's anabolic. Anything that is anabolic is going to upregulate, and anything that is catabolic is going to downregulate. And then just again, quickly to summarize, type 1 diabetes mellitus occurs when an individual's pancreas cannot produce a sufficient amount of insulin. And type 2 diabetes mellitus occurs when an individual's cells do not respond to the insulin produced. Specifically when the glucose transporters, the receptors over there, do not respond to the insulin produced. This picture over here, the pancreatic islets, also known as the islets of Langerhans. We have beta cells over here, and then there are alpha cells and delta cells as well. Okay, and then really quickly, we have glucagon as well. It works with insulin mainly to balance our blood sugar. So we know that when blood sugar is high, our pancreas releases insulin. When our blood sugar is really low, we have the alpha cells and islets of Langerhans in our pancreas, and they secrete glucagon. So when glucagon is released, it's going to increase glycogenolysis, it's going to increase glycolysis, it's going to increase gluconeogenesis, and it's going to increase ketogenesis as well, because it's actually the opposite of insulin. So insulin is going to upregulate all of our anabolic processes, and glucagon is going to upregulate all of our catabolic processes. Because if we are in a starvation period, if we haven't had a lot of food, we really shouldn't be doing anything anabolic. We shouldn't be taking energy and putting it into storage. We need energy to survive, so we have to start mobilizing those energy stores. So that's what glucagon does. It increases anything that is going to help us mobilize these energy stores. So like glycogenolysis, we're cutting off glycogen and we're making glucose. In glycolysis, we're taking the glucose that we just took from glycogenolysis and we're making ATP out of it because we're trying to get that energy. Gluconeogenesis, same thing. We're trying to come up with some glucose so that we can put it into glycolysis. And ketogenesis is just another pathway that we're trying to upregulate our Acyl Coa production. So yeah, these are trends that you should be familiar with. And they are intuitive. So like if you really think about them and you really understand it, you won't need a list in front of you when I say glucagon is released, what is upregulated, what is downregulated. It should intuitively come to you if you understand the concept through and through. Okay, and then we're just going to talk really quickly about a couple of other organs and how they help regulate metabolism. So we're going to talk about the thyroid again. We spoke about it in a couple of other lectures. The thyroid gland is directly responsible for regulating metabolism. It is located in your throat, right below the Adam's apple, right below the larynx. It releases triiodothyronine, which is known as T3, and it also releases thyroxine, which is known as T4. And both of those directly act to increase metabolism. So whenever those are released, they're going to increase all of these processes that we've been talking about. We're going to increase the rate of basically everything. So hypothyroidism occurs when there's not enough hormone created by the thyroid. So our metabolism is slowing. Some of its symptoms are low body temperature, anemia, heart failure is a possibility. The person could be confused, and they will also often gain weight. Because their metabolism has slowed down, their body is not efficiently using their energy stores, so they're going to gain weight. And on the other side of the coin, hyperthyroidism occurs when the thyroid produces too much hormone, which is also not good. So its symptoms are unintentional weight loss because our metabolism is just speeding up. We can't consume enough food for the speed that our metabolism is working at. Rapid heartbeat, again, irregular heartbeat, an increase in appetite, because as our metabolism increases, our appetite is also going to increase to feed it. We could start sweating. A lot of these sympathetic responses are going to be pretty common with hyperthyroidism, and also fatigue. The same thing with confusion. When you're on either end of the spectrum, psychologically, you're not going to feel very well. Mentally, you're not going to feel well because it is a hormonal imbalance. Okay, really quickly again, we'll talk about epinephrine and cortisol, which obviously come from the adrenal gland. We've talked about this a lot at this point. So epinephrine, also known as adrenaline, is released by the adrenal medulla, and it's usually released as an activator for the sympathetic nervous system. So remember that fight or flight. Whenever we are exposed to a stressor, maybe our sympathetic nervous system is going to activate, epinephrine is going to be released, and that's going to act on a bunch of different tissues in our body, and it's going to start all of these sympathetic responses. It also regulates metabolism. So when it's released, it's going to increase blood glucose levels, and it's going to supply the brain with energy during the sympathetic response. So it makes sense that part of the sympathetic response would be to mobilize all of our energy stores, because if we're in a specific situation that we need that fight or flight response to happen, we need to make sure that all of our energy is available as soon as possible. So sometimes you hear these feats. They're probably usually pretty exaggerated about, like, a mother lifting a car off her child, or these superhuman strength, or like these super speed. I think a lot of the reports are exaggerated, but it's because of the epinephrine. So we're mobilizing all of these stores super quickly. So you're going to have a lot of energy to expend in a really short period of time. So that's where that comes from. Okay, and then cortisol is also known as the stress hormone, and it's also released in that sort of situation when we're exposed to stressors. And it's released by the adrenal cortex. The cortex, remember, is the outer part of any organ. The medulla is the inside. So it's going to be in response to external stressors, and it can also increase fat and carbohydrate catabolism. So we're breaking down, again, same thing. We're trying to mobilize those energy stores. It's also going to increase gluconeogenesis, so we have more glucose for us to break down. So all of these conceptually really do make sense. They're not confusing. It's just the nitty gritty and the little steps that are sort of confusing. But like big picture wise, if you really understand it, then you're in a good place. It's just the memorization that is a little bit difficult. Does anybody have any questions about that before we do our last question? Okay, use the chat box if you think of anything. I'm going to start the polls. 1 second. All right, we're live. Give it your best shot, and we will talk about it in a minute. All right, get your final answers in. Okay. Nice, we had a pretty even spread. So 20% said A, 20% said B, 40% said C, and 20% said D, which is interesting. So the correct answer is in fact C. So let's go over it. Which of the following processes would you not expect to see simultaneously as a response to a single hormone signal? So basically, all the question is asking is which pair do not go together. So you're going to have two of them. Each one of them could either be catabolic or anabolic. Which ones don't go together? So if we have an increase in gluconeogenesis and an increase in glycolysis, if we look when released glucagon is going to increase gluconeogenesis, it's going to increase glycolysis as well, because those are both catabolic. So if we look at B, an increase in glycogenolysis and an increase in ketogenesis, again, increasing glycogenolysis over there, increasing ketogenesis, because it's a catabolic pathway. We're trying to break down all of our stores and we're trying to mobilize that energy. Decrease, I'm going to skip over to D, a decrease in gluconeogenesis and a decrease in glycogenolysis. Same thing. If there's increases over here, it stands to reason that decrease would be the opposite hormone. So it's basically the same thing. They're not catabolic, but they're going to be anabolic. Those are anabolic pathways. So C, we have an increase in fat synthesis and an increase in glycogenolysis, which doesn't make sense because the fat synthesis is an anabolic pathway and glycogenolysis would be a catabolic pathway. So you would never really see those two responses from the same hormone. And that's not to say that the two processes aren't happening simultaneously, because all of these things are really happening at the same time in your body. It's just that the fluctuating hormone levels influence how much of the process is going on. So in terms of upregulation or downregulation. So you would never really see both of these processes being upregulated as a result of a single hormone signal, just because fat synthesis is anabolic and it would probably be upregulated by like insulin. And the increase in glycogenolysis is catabolic and it would be upregulated by probably glucagon. Does that make sense to everybody? Cool. All right, so that is the end of everything that I have today. I'm finished for content. Thank you so much for coming tonight. I hope it was really helpful. I'm going to post all of this is actually going to be on the YouTube page immediately, and then I will try to send the PowerPoint in the GroupMe. It's also going to be uploaded to the Google Drive folder as well. I'll stay around for a few minutes if anybody has any other questions, but other than that, have a great night, have a great Saturday, and enjoy the rest of your long weekend. Thanks for coming.